Here.
Yeah.
Dr. Goh is a pediatric neurologist and neuroscience researcher. She has served as associate research scientist and assistant professor of clinical neurology in the Division of Child Psychiatry at Columbia University. She is former co-director of the Columbia University Developmental Neuropsychiatry Program for autism and related disorders. Her research focuses on metabolic disturbances in autism and on the use of brain imaging to identify differences in brain circuits in autism.
Just make sure it's on.
I'd like to welcome Dr. Goh. It's not often that I get to introduce a Rhodes Scholar, so thank you.
It's okay. Thank you so much. It's a real privilege to be here. What I'd like to do this afternoon is to share with you some of the work that we do at my center in San Diego, which is a multidisciplinary autism comprehensive center where we provide a range of different clinical services that, from my research and my time as a clinician in the field, I feel are essential for quality autism care. So I'd like to share that with you today. What does a quality comprehensive treatment program for autism look like? I'm sure you've given that a lot of thought and have done your best to bring together all the pieces for your own child. It's not an easy question, but I believe that it must start with a base in neuroscience. And fortunately, we have several decades now of very good quality in neuroscience, neuropsychological, cognitive psychological research in autism that we know a tremendous amount. And that is the foundation and the foundation of the type of program I'll be sharing with you today. That it's integrated. So my belief is that gold standard care for autism is no longer fragmented care. There are a lot of pieces and they work synergistically, and it's important to have a team that understands and is working actively to collaborate and integrate, and involves community. So it involves a partnership between a professional team and the entire family. I feel it's very important to understand how we got to where we are now. And autism has a very long history. This is a wonderful book that was written in a collaboration between a neuroscientist, Uta Frith, and a historian. And I recommend this to you. It gives a lot of insight into the experience of an individual with autism hundreds of years ago. You are probably all familiar with Leo Kanner, who was a psychiatrist and the first chief of child psychiatry at Johns Hopkins University. And in 1943, he published a case series in which he described and named autism as a condition in a group of children. And since then, there have been a lot of ways in which autism has been misunderstood. And this is a book that was published by a psychoanalyst, Bruno Bettelheim, in the 1960s, in which he stated that autism was caused by mothers, by refrigerator mothers. That particular publication and that theory has caused a lot of damage, in my view, in the area of autism treatment. And this was a documentary that came out and has won several awards in which mothers of children with autism were allowed to now voice their response to that. And so I encourage you to see this documentary. You may also be familiar with the long history of ABA. So ABA, Applied Behavior Analysis, grew out of a field of psychology called behaviorism that was very dominant in the 1960s. These are pictures from a Life magazine article about Dr. Lovas, who was a professor of psychology at UCLA. And you can see that the techniques that have been used in ABA historically have been very controversial. And the field itself is one in which behaviorism is really now viewed as a very specific and very narrow approach to psychology that now has largely been replaced by cognitive psychology and by neuroscience. So I'll talk a little bit more about approaches to behavior intervention for autism, but many of the techniques that have grown out of behaviorism now, in my view, merit revision, because so much more is known about the function of the brain. Now, the American Psychiatric Association publishes what is called the Diagnostic and Statistical Manual of Mental Disorders. And they are the body that defines what mental conditions are. And so over the years, they have defined autism as a set of diagnostic criteria, and that's changed quite a bit over the decades. And this is the current APA definition for what autism is. And you'll see that it's really based purely on symptoms. And it doesn't say anything at all about causation. And I think we're reaching a point in time where our understanding of autism has advanced to the point where this probably isn't the best way to approach or to understand this physiological, biological entity. And much of what I'll talk with you about today has its roots in this publication by Bernard Rimland, which he describes autism as a syndrome and presents a neural theory of behavior. So this book, which just celebrated its 50th anniversary, really is sort of the beginnings of what we understand to be the biomedical, neurological, neurophysiological movement and understanding of autism. And what we know about the causes of autism is very important. So a lot of research has been done, as you know, into the genetics of autism, and we know now that there are many different roots with both genetic and environmental contributions that can lead to the symptoms of autism, and probably close to 1,000 different genes or gene regions that, in complex interactions with one another and with environmental factors, can all lead to a similar phenotype, a similar presentation that we know as autism. And there are many different environmental risk factors. The Interagency Autism Coordinating Committee has done a very nice job of summarizing these research findings. And the risk factors shown here are just some of the ones where the research has now grown, to which there is now consensus that these are all considered environmental risk factors that increase the likelihood of autism occurring. This is a very important review paper published in 2014 in the prominent British journal Lancet Neurology, in which the authors summarize the research findings suggesting that industrial chemicals are contributing factors to neurodevelopmental disorders. So they say, neurodevelopmental disabilities, including autism, ADHD, dyslexia, and other cognitive impairments, affect millions of children worldwide, and some diagnoses seem to be increasing in frequency. Industrial chemicals that injure the developing brain are among the known causes for that prevalence. And to control the pandemic of neurodevelopmental toxicity, we propose that untested chemicals should not be presumed to be safe to brain development, and chemicals in existing use and all new chemicals should be tested for developmental neurotoxicity. And I think what's alarming is that this isn't yet the case, that these researchers have to make a case for this to occur. So, in my view, having been a clinician and a researcher in the field for many years, I think there really is a need and an opportunity now for a comprehensive neurological treatment model for autism. Now, what does that look like? Well, I feel that there are 6 components, so 6 domains of treatment, and these are what we target in the work that we do, and I'd like to walk through each of these with you and share with you what I feel are the most important components of each one. Now, neurophysiology is a term that we use in medicine to refer to the nervous system's electrochemical activity. And this image is a segment of an EEG that gives us a lot of information about the brain's electrical patterns. The brain's electrical activity is highly related to its chemical activity. And so this is an image that shows neurotransmitters which are being released from an axon terminal and communicating to another neuron across the synapse. This is a video just to show you that when we talk about the brain's neurophysiology, it is tremendously dynamic. So there are new connections, new synapses being formed. There are old synapses, old connections being pruned away, and this process is continuously occurring. And this process of network formation, of new connections being formed, other connections being pruned away, this is influenced by so many different factors, by what we experience, by what we learn, through the therapies and the education and the schooling that a child may experience, their home environment, and certainly by all types of physiological, biological factors, including the gastrointestinal health, immune system, and a host of other factors. And what this translates into is how a child perceives the world, their visual function, how they experience the world through emotions, cognition, and then their output into the world, their behaviors, their motor skills, their language. So this is a very complex organ, but we have a lot of different tools and a lot of different ways to influence it, and that's our goal. Now, as part of a neurophysiological evaluation, there are many components. There's a medical history, neurological exam, lab results, electroencephalography or EEG. Our interventions and our evaluation includes understanding a child's response to previous treatments and at times brain imaging. So electroencephalography I'll go into a little bit more depth because there are often a lot of questions about what type of EEG to get, how frequently, and what the results mean. And these are the questions, the next few slides show you the questions that I've been asked most frequently, and I'd like to provide you the answers to. So, does my child need an EEG? Well, an EEG helps to answer many important questions. Is an episode or a type of behavior a seizure or not? Could it be another type of event? If it is a seizure, what type of seizure? And that's important to know because that informs treatment. Are there subclinical seizures? So are there seizures occurring that might not have any outward manifestation? Could seizures be occurring during sleep? So it's very important that an EEG capture sleep. Is there a focal brain abnormality? Is there a part of the brain that's different from the rest of the brain and perhaps should be followed up by brain imaging to take a better look? What is the risk of seizure recurrence if a child has already had seizures? And are there areas of abnormal electrical activity that could be impacting cognition, behavior, or development? So we have a much better understanding now that, indeed, abnormal electrical activity, even in the absence of frank clinical seizures, could still impact development. And then, is a repeat EEG needed? When and why? So what we see on an EEG and how we interpret that helps to inform and answer many of these questions. What type of EEG is best? The right type of EEG really depends on the child. So how long should an EEG be? Well, we know that a routine EEG, which lasts maybe from 20 to 40 minutes, will have about 1/6 to 1/8 of the yield of a 24-hour EEG. So you would need to do 6 to 8 of those to get the same amount of information as doing a 24-hour EEG. So in general, 24 to 48 hours is felt to be the most sort of informative and manageable type of study to get. Is sleep needed? Yes. So it's very, very important that an EEG capture sleep. The purpose of sleep deprivation is generally so that during the EEG the child will sleep. And should an EEG be done in a hospital or at home? Either one is fine, and there are some steps that you can take to help improve the quality of an ambulatory EEG or one that is done at home. And a child's activity during an EEG is not typically restricted, except to say that the more active a child is, the more muscle artifacts there will be on the EEG, which can obscure the brain recording. So in general, a child can engage in their usual activity, but it's best if they can be as sort of at rest and relaxed and comfortable as possible. Video recording is very helpful because it allows the reading epileptologist to correlate the EEG with what's seen on the video. Whatever you can document in the log is also very helpful. Anything that you see as concerning or unusual movement or a lapse of attention. And an EEG is read by a pediatric, it can be read by an individual who has certification as a neurologist. There's an additional certification as an epileptologist that comes from additional training. And my feeling is that for the individual with autism, it's important for the EEG to be read by a pediatric epileptologist because there are very specific findings that those professionals are most well trained to interpret. And then you may be wondering what the difference is between a clinical EEG and a QEEG. So QEEG stands for quantitative EEG, and that's a type of EEG that's done, it's acquired in the same way, but it undergoes an additional analysis and is very often used in conjunction with neurofeedback. Now, in terms of interpreting the EEG, there's information from the background rhythm that can be important, information about sleep quality, certainly epileptiform discharges or seizures are important findings, and all of these warrant very careful attention. So those are some of the things that in the work that we do in my group, we do 24 to 48 hour ambulatory EEGs, and we pay a lot of attention to all of those questions and a detailed interpretation. Now, how common are seizures in autism? Well, we know that in the typically developing population, the prevalence of epilepsy is 1 to 2%, and in those with autism, it's estimated to be 20 to 50%, depending on which research study you cite. So the rate of seizure is much, much higher in those with autism than in the typically developing population. And the prevalence of an abnormal EEG in the typically developing population is 2 to 5%, but in autism has been estimated to be as high as 80%. So there is very clearly a disturbance to the brain's electrical activity in autism. And there are many different seizure types that can occur. They can be generalized, meaning they can involve the whole brain, or they can be focal, involving just one area of the brain. And seizures can appear as brief alterations of awareness or attention, or subtle interruptions to ongoing behavior. So they can be very hard to distinguish between other types of behaviors. So I'd like to show you some videos of different types of seizures. So this first type is probably the one that you're most familiar with. This is called a generalized tonic-clonic seizure, or a grand mal seizure, and involves loss of consciousness and shaking movements of the entire body. This next seizure type is referred to as an absence seizure. And you'll see right here, the child blinks and stares off for just a few seconds. There are a few movements of the mouth, and then he comes back. Okay, that's called an absence seizure. And you can see how that could be very easy to miss. This is a seizure type called myoclonic seizures.
Chase, you're right, buddy?
And each one of those movements there represents a seizure, and these often occur in clusters.
That one was red. You're with Mummy? Okay.
What do you do?
You've just been running around, and then all of a sudden you had one of your episodes again. How are you feeling? How are you feeling? Come on.
That was an example of a seizure type called a complex partial seizure, where the child's awareness is altered, but he's still conscious and he can still respond in part. There were a couple of very classic features that he showed that are very often seen with complex partial seizures. One is that there were some chewing movements of the mouth, and another was that at the end he rubbed his nose. It's not known why, but that's a sort of a hallmark classic feature of a complex partial seizure. Now here are examples of a few atypical movements that are not seizures.
I'm gonna really put this.
So this child has tics. These are called simple tics. They tend to emerge more when a child is relaxed, watching TV or eating a meal. And this is an example of what's called stereotypy, or a repetitive movement, and is very often seen in autism. And those tend to increase during states of heightened emotion, whereas seizures do not. So a very important question in our field has been, in children with autism and epilepsy, could treatment for epilepsy lead to improvement in the symptoms of autism? And for a very long time, the answer was thought to be no, but we now know that the answer is yes. It depends in part on the seizure type, but absolutely yes. The successful treatment for epilepsy can improve the symptoms of autism, and that's why diagnostic testing for seizures and epilepsy is so important. There are several different treatment approaches to try to improve the brain's electrochemical activity, and they include dietary modifications, nutritional supplements, medications at times, and technologies like neurofeedback, transcutaneous vagus nerve stimulation, transcranial magnetic stimulation, and a newer technique called transcranial direct current stimulation. Now, because there are so many different tools that can be used and can be helpful for just this one aspect of autism, it's very important to know how to prioritize them based on testing, which ones are likely to be the highest yield, when to do them, and how to do them, and in what combination, and in what combination with other therapies. So there's a lot to consider when approaching treatment. Just a word about neurofeedback. So neurofeedback is a technique that is often described as exercise or training for the brain. It's a technique in which no electricity or anything else is administered to the brain. So it's non-invasive. Nothing is administered into the brain except for what we call feedback. So it involves an individual looking at a computer screen or a TV where they view a video or play a video game. And what they see on the screen is determined by what's read through a simplified EEG. So they're getting immediate feedback about whether the brain is producing sort of what are considered to be optimal, more favorable patterns of electrical activity, in which case the individual gets immediately rewarded by the image on the screen. The pediatric neurologist at Harvard Medical School, and he says, the literature, which lacks any negative study of substance, suggests that EEG biofeedback or neurofeedback therapy should play a major therapeutic role in many difficult areas. In my opinion, if any medication had demonstrated such a wide spectrum of efficacy, it would be universally accepted and widely used. There are very, very broad applications of neurofeedback in many medical conditions, but also it's widely used for performance enhancement by athletes and executives. Now, a device that's been newer in its application to autism is this one called transcutaneous vagus nerve stimulation. How many of you have heard of vagus nerve stimulation before? Okay, and how many of you have heard of it in reference to an implantable device? So in the US, vagus nerve stimulation is typically done with an implanted device, and that's mainly because of the type of research that's been done and the companies and the marketing that's been done. But abroad, in Europe and Asia, transcutaneous vagus nerve stimulation is more commonly used and has been studied. And there are different ways that the vagus nerve can be stimulated non-invasively. So this is just an example of one device that's available in the US for transcutaneous vagus nerve stimulation. This is a device that stimulates the vagus nerve through electrodes that are placed on the ears. There's a branch of the vagus nerve that runs through the ears. It's called the auricular branch. And stimulation at the ear can, basically through that branch, can stimulate the vagus nerve. Now, the vagus nerve is a major nerve in the body that is responsible for mediating aspects of the autonomic nervous system, which has two components: the sympathetic nervous system, which is the system that mediates the body's stress response or fight or flight response, and the parasympathetic nervous system, which is the opposing system, which mediates the body's rest and digest or relaxation response. And these can be thought of as the yin and yang for the body's stress response. So stimulation of the vagus nerve helps to reduce sympathetic activity and to increase parasympathetic activity. The vagus nerve connects the brain to all of the internal organs, and in particular the gut, and signaling goes bidirectionally from the brain to the internal organs and up to the brain as well. And it's believed that stimulation of the auricular branch of the vagus nerve leads to many different effects in the brain, including impact on the frontal lobes, where it can lead to decreased arousal, decreased agitation. It can have effects on areas of the brain that are involved in emotion regulation, and it seems to enhance a type of EEG rhythm called alpha rhythm that promotes focus and calm attention. Now, another technology that's being used more and more and has been FDA approved for the treatment of adult depression for about 10 years is called transcranial magnetic stimulation. And this is an example of a setup and how the magnetic stimulation is applied. So vagus nerve stimulation, which I just talked about, is the administration of an electrical current, a very small electrical current, to a peripheral nerve. Transcranial magnetic stimulation involves the application of a magnetic field to the brain to induce an electrical current in a focal area of brain tissue. What's very interesting about TMS is that because it's administered in a location in a focal way, you can use it to target specific networks in the brain. And so we are just really at the beginning of understanding the full potential of TMS and how to design protocols that might, for example, target different symptoms of autism. So there are networks, you can see down in this lower right-hand corner, there are networks that we know are important for a function called cognitive control, which is the ability to control one's thoughts and movements. It falls within the domain of executive function. That is thought to be a very important network in autism, and so there are locations of that network that can be targeted through TMS. But there are others as well, for example, language networks, areas of the brain that are important for repetitive or compulsive behaviors. And so there's a lot of potential for protocol development for the use of TMS in autism. John Elder Robison is an author and an individual with Asperger's syndrome who wrote a book called Switched On, a memoir of brain change and emotional awakening that describes his experience of TMS. He received TMS treatment at Harvard Medical School in one of the leading TMS labs, and for him it made a dramatic difference in his social cognition. And so he describes that in his book. There's also a really wonderful podcast from NPR on their Invisibilia podcast, in which a woman with Asperger's describes the effect that TMS had on her social experience of the world and her social understanding. So I recommend that to you.
Experiment.
And there are some very exciting future directions for these therapeutic technologies, including TMS protocols for stimulating language networks, combining therapeutic technologies, for example, TMS and neurofeedback. And there's been one research study so far in autism looking at the combination of those two and has found a beneficial effect in autism. TVNS and neurofeedback and many others. So combinations of technologies really have a lot of potential and are an important area of future research. Transcranial direct current stimulation is a technique that has not been studied in autism, but has been studied in individuals who've had stroke or traumatic brain injury and has shown to be beneficial and to enhance the effects of speech-language therapy. So there's a lot of potential for autism to use devices in combination with non-medical therapies. And then precise individualized protocols based on EEG and on neuroimaging is another important future direction. So let's move on now to think about biochemistry, which is a term that refers to the chemical processes that occur within the body. And metabolism, which refers to the chemical processes in our bodies that convert nutrients into the energy we need, while also getting rid of waste and toxins. And biochemistry is one of the most complex areas of medicine, but if you think about the human body, the cells, and the structures inside of cells, that can really help to give you a sense of what we're talking about, what we're thinking about. This image shows, circled in red, the mitochondria. There are only 2 in this picture, but in any cell there can be up to tens of thousands of mitochondria. And they have a lot of different functions in the cell, including the generation of ATP for energy. So this is just an image of the biochemical pathways, a subset of the biochemical pathways that take place in the mitochondria. And this is actually a 2-dimensional diagram attempting to illustrate all of the biochemical pathways in the human body. So you can see it's incredibly intricate, and there have been attempts to sort of target, very specific aspects of metabolism, but in the end, something this complex is difficult to target in such a specific way. So that's one reason why interventions like dietary interventions and lifestyle modifications are so impactful, because they tend to impact many aspects of biochemical pathways at once. Some of the most important research on biochemistry, metabolism, and autism has had to do with mitochondria. And these are just some of the research studies that have been published showing a disturbance to mitochondrial function in autism. And this was a very important article that was published in 2010 in JAMA, where researchers at UC Davis showed that children with autism had multiple biochemical markers of mitochondrial dysfunction, including reduced function of mitochondrial respiratory chain complex 1, which is just one specific enzyme that functions in the mitochondria. So this research drew a lot of attention to mitochondrial function in autism. And then a couple of years ago, we were able to publish a brain imaging study. This was published with my colleagues at Columbia University, where we were able to show markers of mitochondrial dysfunction directly in brain tissue in children and adults with autism. So this technique, this image over here shows you the technique that we used, where we looked at the brain tissue of these individuals in very high resolution, looking at each cubic centimeter of brain tissue, of which there were about 1,400 per individual. This is the name of the technique that we used. This technique here describes one that was used in previous studies, where instead of looking at the brain in high resolution, the researchers looked in more gross resolution at just 2 large territories of brain, one indicated here by this sort of red slab and the green, and they left a lot of brain areas basically unanalyzed. And so the results of these earlier studies that had attempted to look at mitochondrial function in the brain in autism concluded that there was no evidence for mitochondrial dysfunction in the brain, but you can see that their technique was quite limited. And so when we applied a higher resolution technique, we were able to indeed find elevated lactate, which is a biomarker of mitochondrial dysfunction, and we detected that in 13% of those with autism and 1% of controls, which was a statistically significant difference. And this is just an example of a voxel from one of our autism participants. And you can see here the spectra, and this peak here indicates an elevation of lactate, which is a biomarker of mitochondrial dysfunction. And what these composite maps show you are, when we took all the participants with autism, these boxes show you all the areas in which we found elevated lactate. And the area of brain tissue that was most commonly affected is one shown here. So this shows you, this is the right hemisphere of the brain, and you're looking at the inner surface. And this red region here is called the cingulate gyrus. And it's a region that's very important for the integration of higher order cognition, emotion, and behavior. And this was the area in which we found lactate to be elevated most commonly in those with autism. So we think what we've identified is both a mechanism and also a brain region that for some individuals with autism may explain their symptoms. Now, in this domain of metabolism or biochemistry, there are a lot of different elements that help us to understand a child's profile. And these include the medical history, neurological examination, mitochondrial and other metabolic testing, a nutrition analysis, food and environmental allergy and sensitivity testing, stool analysis, immune and hormonal testing, and genetic testing. And These are some of the markers of atypical mitochondrial function that we look at. This is just an example of an interpretation of a comprehensive stool analysis. And then the different types of genetic testing that we would consider are chromosomal microarray analysis, which looks in very high resolution at an individual's chromosomes to see if there might be extra or missing copies of small segments of chromosome. Whole exome sequencing, which looks for gene mutations or alterations in the sequence of genes. Single nucleotide polymorphism testing, or SNP testing, for what are called common variants, whereas the other tests are looking for more rare genetic variants. There are ways of looking at the genome for common variants that also gives us information. And pharmacogenomic testing, which is a way of looking at an individual's genes that can help us anticipate what medications and the effects those medications may have, what medications may be helpful, and the effects that they may have, and also the potential risk for side effects of the evaluation. And again, there are the same variety and panel and range of therapies that are available in targeting neurophysiology are also available in different ways for targeting metabolism. Now, there are certainly treatment implications for mitochondrial dysfunction in autism. This slide just references some of the research that has been done looking at carnitine, which is a cofactor that helps to transport long-chain fatty acids into mitochondria. And there have been several research studies that have shown a benefit from carnitine supplementation in autism. This slide didn't show it properly, but this is a mitochondrial cocktail protocol that's used by physicians at Kennedy Krieger Institute at Johns Hopkins, and this is a practice parameter that they've published. There's more information here that was blocked off, but you can get a copy of this practice guideline on my website, which I'll give you at the end of the presentation. This is a very helpful approach, a very helpful mitochondrial protocol. This is also a very helpful publication that summarizes some of the major supplements that can have beneficial effect for mitochondrial function and some of the common dosages that have been used and studied. One of the most exciting areas of research into mitochondrial function in autism is being done at UCSD in the laboratory of Dr. Bob Navio, who is a professor of genetics and the co-director of the Mitochondrial and Metabolic Disease Center. He's studying a phenomenon that he has named the cell danger response in autism. And in a moment, I'll show you a research study that we just recently published, but the message that he provides is that autism, which for so long has been thought of as a static brain disorder that could not be changed, may in fact represent a reversible metabolic syndrome. And he's characterized many aspects of what this metabolic disturbance is that's part of the cell danger response. And so he has conducted several animal studies looking at the use of a specific drug called suramin in mouse models of autism, and was able to show that in those mouse models, symptoms of autism and the metabolomic profile improved with the treatment of suramin. This is a small pilot clinical trial that we just completed and published a few months ago, in which we were able to use suramin in a small group of 10 boys with autism, ages 5 to 14. It was a double-blind, placebo-controlled translational pilot study focused on examining the safety and metabolic activity of low-dose suramin. Now, suramin is a very interesting drug because it's been used for over 100 years for the treatment of a parasitic infection called trypanosomiasis. But suramin has many mechanisms of action, including this one that impacts mitochondrial function by blocking extracellular purinergic receptors for the molecule ATP. So our belief is that suramin impacts the symptoms of autism through that mechanism of blocking this very particular type of receptor. And so in our study, the group of 10 boys were matched into 5 pairs and randomized to receive a single infusion intravenously of suramin at a dose of 20 milligrams per kilogram. and the placebo group received normal saline. And we were looking at the outcomes of the ADOS score and measures of language, specifically the expressive one-word picture vocabulary test. And we had several secondary outcomes looking at behaviors and other symptoms of autism. The dose of Suramin was very well tolerated. Some of the children had a very mild rash that resolved on its own. And what we found was that in the group that received Suramin, there was a significant improvement in their ADOS score, and also improvements in language, social interaction, and repetitive behaviors. And those improvements were seen in all of the children who received Suramin and in none of the children who received normal saline. So though this is a very small initial pilot study, the results are very promising, and we're currently in the planning phase for the next study. Okay. So I just want to check how much time I'm already? Okay. So I'll go very quickly through. So those are the main medical domains of treatment that we focus on. Now there are others that are very, very important. And these we might call behavioral, educational, sensory motor, communication, or non-medical. There are new approaches to behavioral, sensory motor, and communication therapies for autism, and they are based on the brain imaging findings that have happened in the past few decades. And I want to share with you this quote, which I think really captures a very important aspect of autism treatment. So E.O. Wilson is an evolutionary biologist and a Pulitzer Prize winner. He was a professor at Harvard for many years. And in his book, Consilience, the Unity of Knowledge, he writes, "The ongoing fragmentation of knowledge and resulting chaos are not reflections of the real world, but artifacts of scholarship." So much of the way that autism treatment has been approached in the past has been based on how we study autism and how our institutions function, and not based on the child and the whole child and the needs of the individual. So now is the time where integrated comprehensive care by a collaborative multidisciplinary team is possible and should be, in my view, the goal. So I'll wrap up by sharing with you, so there are several different therapies that we provide, including music therapy, occupational therapy, physical therapy, speech language therapy, and behavior therapy. And so I want to share, and I'll close with just an activity that I feel helps to illustrate the value of really understanding cognitive processing in autism and allowing that new understanding and the new findings from cognitive research to inform behavior intervention. So this is an activity that I'd like you to do because it illustrates the different networks that we have for visual processing of language and auditory processing of language. And I'll start this video. Can they hear? Are they able to hear? It's good.
Now, I'm okay with lyrics, but I do not know what you're saying there.
Cause I've been acting like sour milk on the floor. Is your...
Oh, do you want us to try again? Okay, great.
No? Oh.
Can they hear?
Sorry.
No?
Okay.
It's okay. And this one?
Now, I'm okay with lyrics, but I do not know what you're saying there.
Cause I've been acting like sour milk on the floor. It's your fault you didn't shut the refrigerator. Maybe that's the reason I've been acting so cold.
Okay, so hang on, go talk me through it.
Cause
I've been acting like
sour milk all on the floor. It's your fault you didn't shut the refrigerator. Maybe that's the reason I've been acting so cold. Cause I've been acting like sour milk all on the floor. It's your fault you didn't shut the refrigerator. Maybe that's the reason I've been acting so cold.
So I hope you were able to hear enough of that to know what just happened. So that was a very complicated auditory processing task that exceeded my capability for auditory processing. And what strategies would help you in that scenario? So children with autism are very often faced with situations like that. And what helps is a visual prompt. Okay? So visual processing is enhanced in autism, and auditory processing is more often impaired. And so the way that we think about how we teach language really needs to shift. And I want to show you here, the words highlighted in green are what we call non-content words. Very often, those words are left out of conventional speech-language therapy programs for autism, thinking that by leaving them out, we are simplifying the task for the child. But these make up the bulk, the majority of words that we speak and the majority of words on a page. And so in our speech-language therapy, we teach these words right from the beginning, because without these words, it's very hard for an individual to understand the language that's spoken around them. So that's just an example of one of the ways that we try to incorporate research findings directly into clinical care as quickly as possible. And I'll move ahead because we're short on time. And I'll close by sharing with you, so we do quite a lot of work with families. Part of our family learning series is to educate families in all of these very important topics. And this is the model of clinical care that really guides the work that we do. So we think about the ultimate goals of treatment and care for autism. And in my view, they include recreation, social relationships, independent living, and occupation. And the foundation for that is really the family and home. And so we focus on nutrition, sleep, the well-being of all family members. We have an amazing team of marriage family therapists and social workers who focus on this element. And we from the very beginning, are focused on neurophysiology and biochemistry and all of the domains that fall within those. And then we look at sensory motor integration, communication, behavior, and cognition, and work on those and have common goals. We then proceed towards lifelong health by continuing our work in the medical areas. And then we try to help a child achieve progress in all these systems of knowledge. We find very often a child can be mainstreamed in school when the right foundation is in place. And of course, all of these things interact. But this framework really helps to guide and prioritize what otherwise is a very complicated treatment process. We have both long distance and local services. And our goal is to provide neuroscience-based, integrated services that build a community and that can be accessed from around the globe. So I refer you to our website, which is www.corticacare.com. We have a center in San Diego, and next month we're opening one in Irvine. And if you feel that there's a need in this area, please go to our website and just send a note to our office, because our third site will either be in Los Angeles or in Northern California. And so if you think that this would be helpful here, please let us know. Thank you.
Thank you so much, Dr. Goh, for such an amazing presentation. I just wanted to mention for those of you who are local, in the East Bay, we have an East Bay network which operates over the WhatsApp group, and we have a whole bunch of classes, groups, meeting groups here for yoga and social groups and music and chanting and all that. So if you're interested, do post on the group or contact one of us, and we can put you on the group so you can get to know what we do here in the East Bay. And there's also one of our yoga instructors who's got a table there, so if you're interested in yoga lessons for your children, please contact him. He does both one-on-one and also group sessions, and he's very good with our kids and very experienced. So with that, I'm